Okay, so welcome to this next video in the playlist on G protein-coupled receptors. In this video, what we're going to talk about is the histamine receptors. Okay, so the structure then for this video, we're going to start by looking at the synthesis of histamine from histidine. We're then going to talk about the 4 different types of histamine receptor, okay, and we will then look at drugs which interact with each of these receptors. So we'll look at drugs which are histamine 1 receptor antagonists, the antihistamines, and we'll look at the drugs which are antagonists and agonists for the other histamine receptors as well. Okay, right. So we'll start off then with the structure of histamine and how we create it from histidine. Okay, so histamine is synthesized from the amino acid histidine. So we'll start off with the structure of histidine and then talk about the structure of histamine. Okay, so histidine is an amino acid, so it has the typical core amino acid structure that all amino acids have. So here is its amino group. Then the amino group comes off the alpha carbon here, which has a hydrogen coming off it. And then the alpha carbon also has a carboxylic acid group coming off. So this is the core amino acid structure that all amino acids have. Okay, now the R group of histidine then has a methylene group in, like so. And then coming off the methylene group, you have a special 5-membered ring called an imidazole ring. Okay, so this is a 5-membered ring where 2 of the members are nitrogens. Okay, and you have 2 double bonds in this ring, one here and one here. So this ring has a special name. It's called an imidazole ring. Okay, and the logic behind that name for this ring structure is that this double bond here between the carbon atom and the nitrogen atom, that's known as an imide bond. So whenever you have a double bond between a carbon atom and a nitrogen atom, that's called an imide bond. Okay, so we have this imide bond here, so that's the imide bit of the name. And also azo, anything with azo in, kind of means pertaining to nitrogen. Okay, so it's quite a nice name because dazole kind of implies that you've got 2, like di, and then azo means nitrogen, so 2 nitrogen atoms. So it's a ring with an imide bond and 2 nitrogens within it. Now all we need to do to complete this structure is then saturate it by putting hydrogens off where there are free bonds, basically. So hydrogens in all of these places. Okay, so that's the structure of a histidine amino acid. Now histidine can be converted into histamine by an enzyme known as histidine decarboxylase. Okay, so this will be done by histidine decarboxylase. And basically, histidine decarboxylase is going to remove this carboxylic acid group off the amino acid and replace it just with a hydrogen coming off that alpha carbon. Okay, so histidine decarboxylase. Okay, right. So let's now draw the structure of histamine then. So we'll have an amino group again, that hasn't changed. Okay, and I'll draw it as much as possible so that it's in the same sort of positions. All the atoms are in the same sort of positions as I put them in this diagram here. Okay, so that you can draw direct parallels between them. So here's the alpha carbon again, and you've replaced the carboxylic acid group just with a hydrogen now. Okay, and then you've still got the R group coming off here. So you've got a methylene group here, and now you've got the imidazole ring again. So this 5-membered ring where we have these 2 nitrogen atoms and this imide bond up here. And now we just saturate everything else with hydrogen atoms coming off it. Okay, so there we go. That's now the structure of histamine. Okay, so a number of cells produce histamine. So for instance, mast cells produce histamine and they store it within special granules. Basophils, which are a type of white blood cell that circulate in the blood. They're a very rare type of white blood cell. You will hardly ever see them on, if you take a blood smear for instance, you will hardly ever see basophils there. They're quite a difficult blood cell to actually see histologically. For another type of cell that secretes them are in the brain. You have certain neurons which use histamine as their neurotransmitter, and there's also cells within the stomach known as histaminocytes. Okay, I'll put this here. So these are called histaminocytes, and these are present in the stomach wall, which secrete histamine. So mast cells, basophils, histaminocytes, which are in the stomach, and histaminergic neurons. These are the four main examples of cells which secrete histamine. Okay. Now, what does histamine actually act on? What are the receptors for histamine? Well, basically, all of the receptors for histamine are G-protein coupled receptors, and these G-protein coupled receptors are the topic for this video. Okay, so they're all going to be G-protein coupled receptors. Now, the thing which links all G-protein coupled receptors together are that they all have this characteristic 7 transmembrane domain structure. Okay, so they all have 7 membrane spanning alpha helices. Okay, and G-protein coupled receptors is the biggest family of proteins in pharmacology, basically. They are an enormous family of proteins. There's over 800 different G-protein coupled receptors. So let me draw out the basic structure. You have these 7 membrane spanning alpha helices like so, and the amino terminus is extracellularly here, and the carboxylic acid terminus of the protein is intracellularly. Now, G-protein coupled receptors can be grouped into 5 separate families. Okay, so the first and absolutely the biggest G-protein coupled receptor family is the rhodopsin family of G-protein coupled receptors. Okay, this contains over 750 of the known G-protein coupled receptors, which is around 800 of the things. So it contains most of the known G-protein coupled receptors. Okay, and indeed all of the histamine receptors are going to be within this family. So this is the one that's important for us. Now, just for completion, I'll talk about the other 4 families. I'll give you their names anyway. Won't discuss them in any great detail. The other 4 families are the secretin family of G-protein coupled receptors, then the glutamate family of G-protein coupled receptors, okay, and the secretin family of G-protein coupled receptors contains, for instance, the receptors to parathyroid hormone, the receptors to glucagon, things like that. The glutamate family of G-protein coupled receptors contains the metabotropic glutamate receptors, and the GABA B receptors are probably the most famous members of that family. The 4th family is the adhesion family of G-protein coupled receptors, and this contains a bunch of receptors which bind to components of the extracellular matrix. And then the 5th family of G-protein coupled receptors is the family of frizzled /taste2. Okay, so this is the frizzled /taste2 family of G-protein coupled receptors, and this is named after the 2 main members, which are the frizzled receptor and the taste2 receptor. And the frizzled receptor is the receptor within the Wnt beta-catenin pathway. The taste2 receptor is a receptor for bitter taste molecules and is used in gustation. Okay, right. So the rhodopsin family of G-protein coupled receptors is the biggest family, and it's the family within which all of our histamine receptors fit into. Okay, now what's special about rhodopsin family G-protein coupled receptors? Well, basically they have very small amino terminal domains and very small carboxylic acid terminal domains. Okay, the characteristic feature of rhodopsin family G-protein coupled receptors is where they bind their ligand. And basically the ligand binds to residues in the membrane spanning alpha helices. So let's draw it here basically. This black dot that I'm shading in here, this is going to represent our ligand. And basically, don't read into this picture too much. I mean, I've drawn it binding to the 3rd and 4th membrane spanning alpha helix, but the reality is that the ligand binds to residues of the transmembrane domains. It's not necessarily the 3rd and the 4th transmembrane domain. Okay, so that's what's special about rhodopsin family G protein coupled receptors. The ligand binds to residues that are in the transmembrane domains. Okay, that's what's special about this family, and most G protein coupled receptors are in this family. So most G protein coupled receptors have their ligand binding to residues within the transmembrane domain. So histamine is going to bind to the G protein coupled receptors, which are histamine receptors, to residues within the transmembrane domains, basically. So let's now talk about the members of the histamine receptor family. Okay, so there are 4 different types of histamine receptor. Okay, so there is the H1 receptor, which just stands literally for histamine 1 receptor. There is then the H2 receptor, which stands for histamine 2 receptor. There is then the H3 receptor, which stands for histamine 3 receptor. And there is the H4 receptor, which stands for histamine 4 receptor. Now, of these, H1 and H2 are the best characterized. H3 is growing. H4 is very, very new. But we do know H4 now exists. Okay, so we have 4 current histamine receptors. Okay, right. Now, we need to discuss which heterotrimeric G proteins each of these histamine receptors is coupled to. Okay, and for this, I would like to discuss with you heterotrimeric G proteins. Okay, just so that you understand when I write the naming system what it actually means. Okay, so basically, heterotrimeric G proteins are these 3-membered protein complexes. Okay, so hetero means different, trimeric means 3-membered. Okay, so these heterotrimeric G proteins are made up of 3 separate proteins that are all different. Okay, so heterotrimeric G proteins are actually made up of an alpha subunit, okay, which I'll draw here. So this will represent our alpha subunit. And the alpha subunit can either have guanosine diphosphate, GDP, bound to it, or guanosine triphosphate bound to it. Now, when it has guanosine diphosphate bound to it, so let's say it has GDP bound to it at the moment, then it's in the off state, okay? And this goes for all G proteins. There is a massive great family of G proteins. Not only is the heterotrimeric G proteins, but there are also the monomeric G proteins. RAS proteins will be the archetypal example of monomeric G proteins, okay? And the feature that all G proteins share is that they are like switches. They have an on state and an off state. And basically, in the off state, you have guanosine diphosphate, GDP bound, and in the on state, you have guanosine triphosphate, GTP bound. So at the moment, it's got guanosine diphosphate bound to it, so it's in the off state. Okay, and let's colour in this alpha subunit in red here. Okay, so here's the alpha subunit of our heterotrimeric G protein. Now, I've drawn it just underneath the phospholipid bilayer. That's what these 2 lines are meant to represent. The inner leaflet of the phospholipid bilayer and the outer leaflet of the phospholipid bilayer. Okay, now what is holding this alpha subunit at the inner leaflet of the phospholipid bilayer? Well, basically it has a lipid group attached to it. Basically, proteins can have lipid molecules attached onto the side of them, okay, so they can have long-chain fatty acids stuck onto them, okay, and this is basically what's happened to the alpha subunit. It's had a lipid molecule attached onto it, and this lipid molecule is anchoring into the lipid bilayer, basically. It goes into the inner leaflet of the phospholipid bilayer, and that's what attaches the alpha subunit to the inner leaflet of the phospholipid bilayer. Okay, now some alpha subunits actually have 2 lipid molecules attached to them and are even better anchored into the inner leaflet of the phospholipid bilayer. Now, these are trimeric G proteins, so we need 3 subunits. We've got 1 at the moment, okay? Now, in the off state, when the alpha subunit has GDP bound to it, then it can also associate with another little complex, and this complex contains 2 separate proteins. So let me show these here. Okay, so this complex is known as the beta gamma complex. Okay, so beta and gamma never ever break apart, basically. There is these two separate proteins, beta and gamma, and they pretty much never break apart. They always stay together. Okay, so often people will just refer to it as the beta-gamma complex, but fundamentally it is two separate proteins bound together. Okay, now the gamma subunit again has got a lipid modification. It's a different type of lipid modification to the alpha subunit. If you know anything about lipid modifications, the alpha subunit is either myristoylated or palmitoylated or both. The gamma subunit is prenylated. Okay. So, it's a different type of lipid modification, but the principle is the same, that you've had this long chain hydrophobic group stuck on the side of you. It anchors itself into the inner leaflet of the phospholipid bilayer and holds the beta-gamma complex underneath the inner leaflet of the phospholipid bilayer. Okay, so we've got these two lipid molecules, and these are the reason that both the beta-gamma complex and the alpha subunit are held at the phospholipid bilayer. Okay, now, when the alpha subunit has GDP bound, then the beta-gamma complex will actually associate with the alpha subunit here. Okay, so here is the beta-gamma complex here. Okay, so beta in turquoise and gamma in pink. Right, so what then happens is when the heterotrimeric G protein becomes active, what will happen is when the ligand binds, a conformational change will occur to the heterotrimeric G protein. These intracellular loops here, okay, this loop between the first and the second membrane-spanning alpha helix, which is, by the way, known as the intracellular loop 1, or ICL1, then the intracellular loop between the third and fourth membrane-spanning alpha helix, which is called the intracellular loop 2, and then the intracellular loop between the fifth and sixth membrane-spanning alpha helix, which is called the intracellular loop 5, those form a heterotrimeric G protein binding site, and they will bind to the heterotrimeric G protein. They then cause the alpha subunit to let go of its GDP, so GDP leaves and the GTP comes in, and once that happens, the alpha subunit, which has GTP bound to it, goes off, and so does the beta-gamma subunit, and they go off and interact with targets. Okay, now, each of these different histamine receptors will interact with different heterotrimeric G proteins. So we need to understand within the human body how many different heterotrimeric G proteins do you have. For this, we need to look at all the different alpha subunits that you have, all the different beta subunits, and all the different gamma subunits that you have. Okay, and this, unfortunately, is quite a headache. Right, so alpha subunits then. Basically, you have 16 different genes for alpha subunits, okay? And by the way, when I say there are 16 different genes in the human genome, what I mean is I am counting the human genome as meaning one copy of each of the homologous chromosomes, okay? So in the nuclei of all of your cells, so where should I put this? Okay, so let's have a cell down here. In the nucleus of all human cells, of course, you actually have 46 chromosomes. Now, of course, these are paired together, so in fact, you actually have 23 pairs. So, for instance, you have 2 chromosome 1s. Now, they're not identical. Of course, they will have different sequences of organic bases, but the genes on them will produce the same sort of protein, basically, a protein that does the same function. Yes, they might not produce identical proteins, you know, they might have slightly different combinations of amino acids, but they are similar enough that they do the same function. Okay, so basically you have 2 homologous chromosomes of each type. Okay, when I say the human genome, what I mean is just take one copy of each of the homologous chromosomes. So really I mean take one chromosome 1, take one chromosome 2, one chromosome 3, all the way up to one chromosome 22. Okay, and then we'll ignore the sex chromosomes on the end. So within that sort of karyotype, as it's called, so your chromosomes 1, 2, 3, all the way up to 22, 2, okay, up here, basically you will have 16 genes for alpha subunits. Okay, so in your full set of chromosomes, you'll obviously have 32 genes, because on all of the homologous chromosomes you'll have another copy, but I'm not counting that, basically. So when I say you've got 16 genes, I mean if you look through one copy of each of your homologous chromosomes, you will find 16 different genes which all code for proteins which can function as the alpha subunit of a heterotrimeric G protein, but all have slightly different effector functions. So when they're activated, they will trigger completely different, completely different outcomes. Okay, so it gets more complicated, because although there are 16 genes, there are actually 21 known alpha subunits. Okay, so what you might say is, oh, does that mean that some of the genes aren't, we haven't found some of the genes for some of the subunits? Well, the answer is no. We have found the genes. The reason there are more subunits than there are genes is that for some genes there are alternative splice variants. Okay, so let me just remind you of the central dogma of biology. Okay, so if we have our gene here, so let's say this is a gene for the alpha subunit of a heterotrimeric G protein. Okay, and you should know, of course, that the DNA has to be transcribed into a piece of mRNA. Okay, so we can divide the two strands of DNA into two different pieces, basically. One of them will be the coding strand, so let's say this pink one will be the coding strand, okay? And this green one is then the non-coding strand. Now, what does this mean? Well, basically, when you transcribe the DNA, you do not need both strands. What you do is you create a piece of mRNA that is complementary to one of the strands, and the strand that you pick to make it complementary to is the coding strand. Okay, so here is our piece of mRNA in orange here, and it's complementary to the coding strand of the DNA, basically. Okay, and this mRNA is then called the primary transcript. So it's an exact complementary copy of our coding strand, or if you like, it's an exact copy of the non-coding strand, except for the fact, of course, that we've replaced thymine with uracil, because this is RNA and this is DNA. Okay, so this is what's called the primary transcript. Now, the primary transcript is not what you put through a ribosome and get a protein out. So you don't send the primary transcript straight off to the ribosome to be translated. No, instead what you do is you first cut bits out. Okay, so here's the primary transcript. Basically, the primary transcript isn't ready to be translated yet, because there are loads of bits that don't actually need to be there when we translate it, and if we did translate it with those bits in, we'd end up with a protein that was just ridiculous. Okay, so the portions that you actually want to keep in this primary transcript are known as the exons. These are the portions which actually code for useful pieces of protein, basically. So these 4 pieces that I've shown here, these are meant to represent exons. And the portions in between, basically, here and here, these are then known as introns. Okay, so you have exons with introns in between them. Now, basically, what's going to happen is before the primary transcript actually goes through a ribosome, what you're going to do is you're going to cut out the introns. So you're going to cut the RNA up, and then you're going to stick the exons back together, and this process is known as splicing. So what happens first is the primary transcript is going to go through splicing before it actually is sent on for translation, and then the mature piece of mRNA, okay, as it's called, which is the exons re-glued together, will then go through the ribosome and be translated into a protein. Okay, now, splicing basically involves cutting the mRNA up and then re-stitching it together. Now, basically, there are different ways that you can obviously do this. So, Sometimes, a quite extreme example is, for instance, sometimes there are exons that either you can include or you don't have to include, i.e. it's a choice, it's voluntary. If you include it, you'll get a bit of a longer protein, which will have a slightly different structure. If you don't include it, you'll still get a functional protein. It'll be slightly different, but it's still a functional alpha subunit. And that's quite an extreme example, where there are whole exons that can be included or not. Alternatively, there might be exons where it's not necessary to have the entire full portion of the exon, i.e. you can cut it at slightly different regions basically, and you'll end up with slightly different proteins depending on where you actually start and finish the exon, but it will function in the end. Okay, so often there are different ways that you can splice the primary transcript to end up with a mature mRNA. So basically, from the same primary transcript, you can get multiple different mature mRNAs, which will then produce different proteins. And this is how one gene can produce different proteins. Okay, and these different proteins that are produced by a single gene are called the splice variants of that gene, or the splice variants of that protein. Okay, right. So that's how we go from 16 genes to 21 alpha subunits. Basically, some of the genes have multiple splice variants, so it's not just some bizarre anomaly here. Okay, so let's now talk about the different alpha subunits for heterotrimeric G proteins. Okay, so they are classified into certain families. Okay, so there is the G alpha S family first. So there's 4 families into which the alpha subunits, the 16 genes of the alpha subunits, are classified. Okay, so there's the G alpha S family, which contains 2 genes. Okay, it contains the G alpha S gene and also the G alpha OLF gene. Now the G alpha S gene has it has quite a few splice variants, okay, so it gives rise to more than one protein in the end. G alpha OLF doesn't have multiple splice variants, at least not that we know of. Okay, then the next family is the G alpha I/0 family, and this is the most complicated family. Okay, so this family of alpha subunits of heterotrimeric G proteins has 8 members. Okay, so there is the G alpha I1 subunit, there is the G alpha I2 alpha subunit, there is the G alpha I3 subunit, and then it goes on, so those are the 3 I subunits. There is then the G alpha T1, okay, and also the G alpha T2. Now these are the alpha subunits that you use in transducin, which is a heterotrimeric G protein used in the photoreceptors in the eye. Okay, G alpha T1 is the alpha subunit that you use in transducin in rod cells. G alpha T2 is the alpha subunit of transducin that is used in cone cells. Okay, let's continue on with this family then. So I'm just going to go over here so we can fit it all in. There is the G alpha O alpha subunit, there is the G alpha Z alpha subunit, and then finally, finishing off this family, there is the G alpha GUST alpha subunit, okay, which is used in the gustatory sensory system. Okay, right. We're not going to fit all of the alpha subunits on here, so let's try though. Let's do the G alpha. The next family we're going to do is G alpha Q/11. Okay, and this has 4 members in. Okay, so remember I said there were 4 overall families basically. We've done 2 of them now. Let's do this 3rd one. Okay, so the G alpha Q/11 family contains the G alpha Q alpha subunit, the G alpha 11 alpha subunit, and then also G alpha 14. And then this final one's a little bit more complicated. Okay, so this is sometimes called G alpha 15/16. Okay, but this is not 2 separate alpha subunits. So let me just go over the page and discuss this. So basically, if you do your research in mice, you will find an alpha subunit that is called G alpha 15 if you do your research in humans, you will find an alpha subunit that is called G-alpha 16. Okay, so this is the murine subunit, meaning that it's found in mice, and this is the human subunit. Okay, now these two effectively do exactly the same thing. This is the human version of G-alpha 15. So, some people will just say that humans have G-alpha 16. Some people will call this instead G-alpha 15/16 because effectively it's the same thing as we found in mice and had called the G-alpha 15. So humans definitely do not have a G-alpha 15. Their equivalent is the G-alpha 16, but some people will have said let's rename G-alpha 16 G-alpha 15/16 to realize the fact that we called it 15 in the mouse and called it 16 in the human. So effectively we should have used the same name for both of them, and this is to try and compensate for that later. But you will sometimes hear it referred to as G-alpha 16, and then if you're reading something about the mouse, they might call it G-alpha 15. If they're being nice, they should call it G-alpha 15/16. Okay, so that is the fourth member of the G-alpha Q/11 family, and then the final family of alpha subunits is the G-alpha 12/13 family, and this just contains two members, G-alpha 12 and then also G-alpha 13. Okay, so we've now got two of these G-alpha subunits here. We've got 4 in this family, 1, 2, 3, 4, that takes us up to 6. We've got 8 in this family, that takes us up to 14, and 2 more here, that takes us up to 16. So those are all the 16 genes that are coding for alpha subunits of heterotrimeric G proteins. Now we'll continue this discussion in the next video where we'll talk about the beta and the gamma subunits.